So my name is Edmund. I'm the Immunology Sales Manager in ProImmune. I'm based in Singapore, and I oversee the company's activities across the Asia-Pacific region. So I graduated from National University of Singapore and began my career in Abbott Laboratories, and then conducting research on the impact of mineral fortification on the aggregation of dairy protein. And ever since afterwards, I joined ProImmune in 2019, and has been part of the pivotal in terms of supporting both academic and industry partners in addressing immunological challenges associated with biologics and therapeutic development. Through close collaboration with clients, so I have contributed to advanced research strategies and enabling successful outcomes across a diverse range of projects. So my work has been focusing on bridging scientific insight and practical application to support innovation in immunology and drug development. So in my talk today, I'll be sharing the technologies that ProImmune is able to offer in what we call an integrated approach to adaptive immunogenicity assessment that can be actually implemented in both biologics and also in peptide therapeutics. That's where I'll be focusing on. So my name is Edmund, and I also have a Chinese name, obviously. And my name is also, you can call me Liang Guorong. So feel free to reach out to me if you want to, we can converse in either Chinese or English. I'm both comfortable in it. So as a form of introduction of our company, ProImmune, so the question is, who are we? So we are considered as your strategic partner that we can help you fully characterize and to understand the wanted and unwanted immune responses using our state-of-the-art proprietary technologies. So we are recognized as a world leader for providing such products and services for more than 25 years in the area of preclinical and clinical immunology research. So you might think that it's kind of like a little bit thick skin that we consider ourselves as a world leader, but actually instead it's the conversation that we have over the years with customers that we have been engaging with. They have been mentioning about us, recognizing us, that we have the knowledge and the tools that we are able to support and achieve the goals of their project. So we are serving almost all the expert immunology groups in the academic space. So that's actually how the company started off about 26 years ago by my CEO, Nikolai Schwab, whereby we first started off the companies providing the MHC multimers product with the MHC class 1 multimer product, allowing you to actually track and enumerate the antigen-specific CD8 T cells. And further on, we then move on to develop services, assays, that allows us to actually better characterize responses, especially focusing on looking at T cell responses to identify the response as well as the identification of the epitope. So that's where then we further on expanding the services that we have now, services whereby we are supporting companies in the biotech and the biopharma industry in terms of focusing a lot in the area of immunogenicity assessment for biotherapeutic drugs and also peptide therapeutics. So just showcasing some of these academic groups, universities that we have been working with them for many years, and also some of the companies that we are also working with. So just give you a quick overview of a quick image of where our company is located. So our headquarters and our lab operation, they are all based in the Oxford, UK, and we have sales support based in the US as well as in Singapore. So myself and my colleague Wen-Zhe Chua is based in Singapore, supporting the whole of Asia Pacific for ProImmune business. So in terms of mastering immunity, so that's the theme of this meeting. But in order for one to actually master immunity, we need to first understand that it's a very complex topic. I think most of us will agree on this. So as you can see, in the understanding of the immune response, we will need to understand the antigen, how is it being processed, the restriction of the peptide binding to the MHC, understanding whether or not it's a T cell epitope, able to actually drive a T cell response, and this is the adaptive side of things. What about innate response? That's something that is very critical to be addressed in the process of understanding the risks of your therapeutics, whether or not does it actually going to be inducing innate immunogenicity that could lead to cytokine release syndrome. And we also need to think about the design of the protein in terms of the variants, the engineering of the sequences. Is it going to be engineering a new epitopes into the sequence, potentially going to be driving a T cell response that could lead to downstream immunogenicity ADA production. And also, ultimately, we need to think about the B cell function as well. So, in today's talk, I will be focusing on these 4 assays that we offer them as a service. And more often, like what previously Dr. Tim Hickling has mentioned, in order for us to understand the overall risk of immunogenicity, more often than so, you would need to combine several assays for us to really understand and characterize the risk of your biologics that you are developing. So the first assay that I will be going through will be focusing on the ProPresent antigen presentation assay, the ProMap T cell proliferation assay, the ProScern DC T cell proliferation assay, and then lastly with the review MHC peptide binding assay that I'll be focusing on in today's talk. So before that, I think in the room we can agree that immunogenicity is very critical and it can lead to unwanted immunogenicity that can lead to treatment failure and also will then lead to the project in terms of discontinuing. It can be that critical, and there's a lot of evidence, there's lots of papers out there that has been describing this. So it is very critical for us to understand this in the early stage, as early as possible, as what previous Dr. Tim Hickling has mentioned, to really understand where are the risks and how can we actually remove liabilities design in the design of your drugs that you are working on. So in terms of biological drugs, peptides, consumer products, and even food ingredients that we are eating for those proteins that we are having, and they contain novel protein sequence content that will be recognized by our immune response. So for immunogenicity, it's driven by a complex set of factors. So what I've done here is to narrow down into 2 different factors that we can consider. So it's categorized in extrinsic and intrinsic. So extrinsic, in most of the cases, it's not something that we are really in control in the early phases of the design of your drug development. In terms of the route of administration, frequency of exposure, the patient, that's not something that we often can have access to in terms of the knowledge in the early stage of the development. So more often, where the focus are in the preclinical phases are usually the intrinsic factor, where we focus more on the amino acid sequence, the formulation, the aggregation level. So those are things that we are able to control and we can understand better in terms of, let's say, the sequences. Are we able to actually remove any kind of liability from the sequences for us to reduce the overall risk of the development of the drug? So demonstrating these, a lowered kind of immunogenicity or equivalent antigenicity of your drug, this gives you a significant competitive benefits against the other companies that are working against similar targets. And then in terms of managing immunogenicity risk by design, it's essential to ensure a therapeutic reaches market without any competitive disadvantages. So this is really key, whereby companies, there are targets that are very popular, such as, like, I can remember there was a lot of conversations having in the PD-1 space, the PD-L1 space, and then there was a lot of company getting into it. So the question here is, how can you differentiate yourself from your molecule that you're working on, the antibody against PD-1? So many companies are working on it. How can you further differentiate yourself that your molecule is better than the other company? So immunogenicity does plays a very important role on how you can actually differentiate your antibody to your competitor. So now the question is, now we have established that immunogenicity is a critical part of your drug development journey. So the question here is that how can we mitigate adaptive immunogenicity risk in the early stages of your development? So I think first of all, we need to establish the pathway. So this is a very simplified cartoon on the pathway in terms of immunogenicity. So let me take you through this. So first of all, we have the biologic. It could be a peptide drug, it could be an antibody, a bispecific, an ADC. And then what happens next is that when it gets into the body, it will get uptaken and then processed and presented by the antigen presenting cells, which is the APC. And then followed by the presentation through the MHC class II molecules, which is this blue color circles here, followed by this red color line, which is the peptide that is being presented, and then this peptide that is actually derived from your protein molecule in this case. And then this is where the CD4 T cells will start trying to recognize through the TCR to bind to the MHC class II molecules, and if it's able to bind to it, it will then start proliferating, secrete cytokine, activating the B cells, and then release the anti-drug antibody that is actually able to recognize your molecule. So this is where immunogenicity of this ADA, there's lots of different effect onto your molecules, such as like loss of efficacy, altered PK/PD, or even more serious, it can cause cross-reactivity to your self-protein that could lead to autoimmune disease for immunogenicity implication. So now the question here is, where are these assays that we can actually deploy in your preclinical phases to really understand where are the risks for us to better mitigate these and to avoid what happens in the downstream of immunogenicity of the ADA production? So where the assays focus on is a lot in this area of the mechanism of the interaction between the CD4 T cells targeting the MHC class II molecule. And the reason why this is such an important pathway of the mechanism, because these are helper T cells that help in the maturation of the B cells to release high binding affinity antibody that can target your molecule. So that is why we could actually understand this better, where are some of the risks here, we could actually remove the downstream, downstream responses of the immunogenicity liabilities. So we have the ProPresent MAPS assay, where we focus on the presentation of the peptides, and then the ProMap T cell proliferation to understand the function of the CD4 T cell, whether or not are they able to proliferate. And we have the DC T cell proliferation, whereby it's a whole protein assay where we load the whole protein to understand whether or not it's a functional T cell response exists. So just moving back for the T cell proliferation wise, this is actually a peptide-based assay where we load usually a 15 mer peptides directly to the PBMC, followed by measuring the function of the CD4 T cell proliferation. So just to differentiate between these two functional T cells, between the T cell and the DCT assay. And finally, we have the HLA binding assay, where we will then understand the peptides against a set of HLA allele, whether or not what is their binding characteristic, what is their stability of them when they bind to the MHC molecules. So first of all, running this assay, we will need to, of course, have cells. So in ProImmune, we have a biobank where we source our donors from the UK, isolate the PBMC, and then we perform a HLA typing, and then we'll cryopreserve them at our biobank. And once we initiate a project, we will then use them from our biobank to run the respective assays, such as like the ProPresent T cell assay and also the DC T cell assay. So the good thing about that in the UK is that in the Oxford especially, is that we do have a variety of ethnicity groups that in the population. So the source of the donors is kind of a wide variety in terms of ethnicity. So it's quite a good coverage that we can have in terms of the HLA typing that we, the donors that we get. So the coverage that we have in terms of running such assay is typically covering the global distribution as much as possible in order for us to really understand where are the risks in terms of which HLA there's a higher risk, and for us to have actually a macro perspective in terms of understanding in the diverse range of HLA alleles, where are some of the risks that in terms of the immunogenicity of your molecule. So using human PBMC, we know that they are clinically relevant immune response for them to use for our assessment. And then we have a diverse donor pool covering broad global HLA allele representation as mentioned. And let's say if you want to focus on a certain region for your molecule, let's say for example getting into a certain market, we are flexible in terms of selecting some of the HLA alleles to represent the target market that you want to get in, and we can work on that with you to help you customize the donor panel that you want in the assay that you want to perform. And then in terms of using these PBMC donors, we will then use them in the antigen presentation, the ProPresent, and also in our T cell and DC T cell proliferation assay. So the first assay here on the ProPresent antigen presentation assay, it's an assay that is very powerful that allows you to identify naturally processed and presented epitopes. So this assay is able to apply and deploy across biologics, peptide, gene therapy vectors, vaccines, and immunotherapies. So in today's case studies that I'll be sharing, it's more towards the biologics as well as also or some other modalities that will be covered in this talk as well. So for this assay, we are able to perform the class 1 analysis as well as the class 2 analysis, but for today's context for immunogenicity, we'll be focusing more on the class 2. That's the pathway in terms of driving the CD4 T cells from the presentation of the peptide through MHC class 2. So in terms of the turnaround time that we are able to test multiple test articles, and it was discussed earlier in terms of like the throughput of the assay. So this is really driven by the amount of cells that is quite intensive, using a lot of cells for this assay. So that's where typically we usually would recommend in terms of like the cells usually testing 4 proteins at max, as this actually kind of like the resolution of the assay depends on like what is the starting amount of cells that we start with the assay. So this is something that can be adjusted in terms of how we can actually perform the ProPresent assay. So the turnaround time of this assay is around 4 to 6 weeks, and then this allows you to actually supporting your project for making an efficient decision. So typically how this assay works is that we will start off with your protein, we'll load into the monocyte-derived dendritic cells, allow it to uptake, process, and present that on the MHC class 2 molecule, and then we'll perform an immunoprecipitation step to actually pull down the MHC class 2 molecule loaded with the peptide that is derived from the protein, and then we will then pull down the peptides and put through the tandem LC-MS-MS, and followed by sequencing what are these peptides that has been presented through derived from your protein molecule. So this is how the data set will look like as an example. So from here, I can take you through. So these are the donors. These are individual donor IDs. And then these are the HLA earlier. This is focusing on just the HLA-DR analysis. And this is looking at the testing the adalimumab for this case. So this is where you will see more often in terms of the peptides that are being presented. They are actually overlapping nested set of peptides that you will see them repeatedly presented by each of the donors. And each of these sequence that you see here are actually unique identification of this peptide that is being presented by the donor. So then what we do next is that from here, we will then summarize up the peptides that has been identified across the panel of donors, and we will then identify which of these peptides they are actually promiscuously presented in a way whereby if they are presented across multiple donors, it means that there is actually a relatively high risk that it could be actually a T cell epitope that can actually drive a CD4 T cell response. So in this case, peptide position 74 to 92 is promiscuously presented, and because it was identified in all donors from these adalimumab sequences were detected, as you can see from the third row here for this sequence that you can see. So these numbers are representing the number of copies that we have identified through the mass spec analysis. You can see across that all donors have actually presented this peptide. So the next case study here, which Dr. Tim Hinckley has briefly mentioned about this Pfizer project for anti-IL-21 receptor, a fully human and therapeutic antibody. So in terms of this, they went on into the clinical phase 1 study. They developed this fully human anti-IL-21 receptor monoclonal antibody, and they've seen a 76% ADA rate in their clinical study. So then they asked the question in terms of, where are the risks in terms of the sequence of the antibody. So what we propose here is that we perform the ProPresent assay in terms of identify where are the T cell epitope, and you can see from this data set here. So the first row here is the overlapping of the donors of the peptides that are being presented, and you can clearly see that there's a hotspot region at the CDR2 light chain area where multiple donors are presenting peptides from this region. But the question here is that presentation of peptides doesn't mean that it's actually a T cell epitope that is driving the CD4 T cell, because we need to bear in mind that this is antigen presentation assay that allows you to understand the presentation of the peptide, but it's not a functional assay. So that is where they perform the second assay to understand whether or not this is actually driving the CD4 T cell response by performing a DCT assay. So this is a very nice example where you need to rely on the combination of assay to really characterize what is the risk of the molecule that we are working with and to understand whether or not is that actually a problem in the molecule. And another point that was actually mentioned in this paper is that about the in silico prediction was over-predictive. So that is where we can look at the data below. So Pfizer done the in silico prediction across the antibody, and you can see that the hotspot regions come out in many different parts of the antibody. So then the question here is that if it's coming out in so many parts, does that mean that we need to work on each and every region to remove the liability? We need to understand that in silico has this inaccuracy, so we need to rely on physical in vitro assay to really understand where are the real problem. And so rightly show that in the ProPresent assay, there is one key spot where things needs to be sorted out in terms of removing the liability, and this is where the region was validated as the problem in the sequence of the antibody. Yeah, so there's an over prediction in the in silico assay that was being performed. The next case study here, it's a CRISPR-Cas9 immunogenicity. As we know that CRISPR-Cas9 is a Nobel-winning prize, and there's a lot of opportunities, unprecedented area where it can offer in the gene therapy area. So I think the first question that comes to mind, especially I think from the regulatory perspective as well as also as a drug developer, is that what is the risk of immunogenicity, especially that we know that Cas9 protein, they are bacterial origin, and if there were to be any, unwanted immune response from the Cas9 protein, that is going to be a very serious implication. So that is where this paper that's performed by Editor's Medicine with the FDA, they co-published this paper, and they identified 22 Staphylococcus aureus Cas9 peptides that were both presented on the MX class 2, so that's the first part, as well as they are able to stimulate CD4 T cells. So what I want to show you here, you can put a focus on the right-hand side of the plot here. So what we are looking here is that the pink color boxes here, these are regions of the Cas9 protein that has presented peptides and was identified from the ProPresent MAPS assay. And then followed by the colored lines here, and okay, sorry, each row here represents a single donors. And then for these lines here that you are seeing, these are sequences that are capable of inducing a CD4 T cell response. So basically what you can do here to understand the risk of the region of this protein is by looking at the overlapping region of the pink boxes with the lines, and that is where peptides are capable of presenting and also capable of inducing a CD4 T cell response. So as what we can see here, almost 85% of the identified presented peptides are able to induce a T cell proliferation. And the next case study here is Vetrocorp alpha, which is an engineered factor 7A analog. So this is a work we have done with Novo Nordisk, where we know that recombinant factor 7A has been used for more than 20 years without any report of immunogenicity. So what Novo Nordisk wanted to do is to engineer a better version of the recombinant factor 7A for better enzymatic activity. So they introduced 3 mutations to the factor 7A protein. So at that point, he was thinking, they are retaining 99% homology of the protein. So potentially, this is actually maybe a low risk that they are not really looking too detailed about this engineering of these sequences to the protein. So when they enter into the clinical phase 3 trial, even though they were effectively treating the patient, but because of this treatment is a last line of treatment for hemophilia patient, having ADA production was actually considered critical, life-threatening. So ADA inhibitors was observed in some patients at phase 3 trial. So then they had to actually discontinue this late-stage drug development in the later stage of this program. So then Novo Nordisk then had to come up with the question, approached us and asked, how can they ensure that neoepitopes are not engineered in leading to development of ADAs? So then we designed a 2-stage approach to address this question. So first of all, we suggested, let's run a ProPresent assay to identify where are the regions that are being presented. So that's the first question. So we run a parallel study with Novo Nordisk. So on the left-hand side, Novo Nordisk ran a parallel study for antigen presentation assay, and followed by ProImmune, we ran parallelly with Novo Nordisk using our ProPresent assay. So I just want to bring your attention to two key points from this slide. Is looking at the VetraTop alpha, the regions that was identified from Novo Nordisk and ProImmune, they were similar in the regions that was presented. And the second point here, this presented region for this peptide contains 2 out of the 3 mutation. So then now the question here is actually these 2 mutation that has actually caused the failure of their development. So we can't jump into that conclusion yet, because we need to remember this is just antigen presentation. We need to then confirm, are they triggering a T cell response? So that's where the stage 2 of this assay is, whereby we synthetically synthesize those peptides and then we incubate them into the PBMC, running a T cell proliferation assay, incubating for 7 days. And this is where I want to bring your attention to highlight the data here. So we synthesize the peptides that covers the wild type sequence as well as the variant sequence. So you can see that for 158, the blue color bar here is the wild type, and then the green color bar is the mutant sequence. So you can see that there isn't, not too much of a difference in terms of the T cell response, and it wasn't identified through the MAPS assay as well. So if you look at the position 296 and 298, and bearing in mind that they were actually identified through the MAPS assay, you can see that the response that we are seeing, it's almost 2 times as compared to the wild type. And what we did further was to actually synthesize peptide that contains both mutation, and you can see that the response was even higher. So if you take a step back in terms of the preclinical phases, to think about it, if this assay were to actually be deployed in the early stage of selecting the variants of your sequences, the design of your protein, I would think that very likely so, that we would not be choosing this amino acid as part of the engineering. We probably might choose another candidate of sequences to engineer the protein and to improve the overall design of the protein with this sort of important and critical data set that can actually allow you to make informed decision for your design of your therapeutic. So this brings us nicely to our next assay, our ProMap T cell proliferation assay, which is a peptide comparative analysis, where we use CD8 depleted PBMC, and typically we will run a 40 donor study, but we are flexible in terms of the number of donors that you want to perform, and that really depends on which stage you are at for your study, which this can always be discussed in more details. And we type them for DR, DP, DQ for their HLA class 2, and typically how we actually select donors for the study, they are representing the global HLA frequency. So you have a good coverage of the different donors as more often you want to actually bring your drugs to multiple different regions and not just focusing on just one market. So we can cover those global distribution. So in terms of the study design of how do one select the peptides to be tested in the T cell assay? So one can actually do by doing an overlapping strategy by using 15 more peptides covering a certain region, let's say the CDR2 region. You want to do an overlapping sequence offset by 3 amino acids. We can then synthesize the peptides. Or you can do a targeted screening like what was described earlier in the case study with Novo Nordisk. By running the ProPresent, identify the peptides where are some of the potential liability. We will then synthesize them as synthetic peptides, and then we can run the assay using those peptides. Or you can also use an MHC peptide binding assay to understand where are some of the peptides that are binding very strongly and stably to the MHC molecule, and from there you can then select the peptides that you want to actually screen in the T cell assay. And also this design for the T cell assay, we can also test for peptide drug impurities with potential T cell epitopes, and this is very applicable especially for generic peptide, complex generic peptide projects, where when you synthesize a peptide, there are modification to your peptide in terms of insertion, deletion, this sort of things in the impurities profile of your peptide drugs. This is where the T cell assay has been very useful in assessing the peptide drug impurities as part of the submission to the regulatory agency. So how this assay works is that once we synthesize the peptide, we will then incubate the peptide 7 days into the PBMC, and they are CFSE labeled, and then we can then use flow cytometry to identify the proliferation of the CD4 T cells. So this is typically how the data set will look like for the T-cell assay. So on the left-hand side, you can see that these are our assay positive control, which comprises of tuberculin PPD, KLH, and 2 peptide pools, which is the CFT peptide pool and KLH peptide pool. And you can see a nice and strong positive response from the positive control. And just to highlight that each of these colored boxes represent a single donor. So basically, this is actually a 40-donor study. And for the rest of these peptides here are the peptides that you want to select to actually test them in this assay. We will synthesize them. And then from here, in terms of understanding the assay, for the data, it's a relative antigenicity of, let's say, for example, you have multiple variants that you actually want to test. And then from there to select the best design, the most optimal design that you want to be included in the design of your antibody. So let's take an example between peptide 3, 4, and 5. So let's say these 3 variants are things that you want to select and they're all coming from the same region. So from here, you can do a very simple relative comparison between peptide 3, 4, and 5. And from here, peptide 5 with just 1 donor that is actually coming up as a positive signal. So this is how you can actually decide on which has the lowest risk of driving a CD4 T cell response as a risk of immunogenicity for your development of your protein drug. So then, what about complex generic peptides, as well as what are some of the regulatory expectations? So many years we have been interacting with multiple companies in the space of complex generic peptides with the FDA coming up with the guidance, where the ANDAS guidance, where you are able to get a waiver from the clinical trial if you are able to actually demonstrate using in vitro assay in a very stringent way that your profile of your generic peptide is actually comparable to the RLD. So this is where some of these key expectations after many years of learning through the process of interaction with customers as well as regulatory agency. So the testing of peptide-related impurities can potentially T-cell epitope, and that's the reason why we need to understand the impurities. Are they driving a T-cell response? And then this is where we need to assess individual impurities using the T-cell proliferation assay or using the HLA binding assay. And we focus on impurities that has profile between 0.1 to 0.5%. And whether or not they are novel or present in a higher amounts as compared to the RLD. And typically, you will need to consider at least a 40-donor study in order to have a good coverage during the submission to the as part of the data package to the regulatory agency. And more importantly is that in the study design, overall study design of the T-cell assay, we need to consider having the suitability control and then using your generic peptide API as well as the purified individual impurities and testing them at equimolar concentration. And this concentration should not be impacting the APC cell viability. So this is where, in terms of the assay, we have to design in terms of having the suitability control. And I remember having this conversation with multiple customers and also with the regulatory agency when the comments coming back is that we need to design the assay to include additional controls, which is called the suitability controls. And we were tasked with this challenge that this single peptide needs to be promiscuously binding to multiple HLA allele. And we were start to think, where can we actually find such peptides? So we've gone through the process, thinking about multiple different options, thinking about Padre peptide, is it suitable? And then in the end, we then tested a lot of different options of candidates to see which of them actually perform most consistently and able to actually give you a moderate level of response in the T-cell assay. So this is where after several rounds of testing, so we identified that exenatide and samocalcitonin were able to give us a consistent data and a moderate response from the T-cell assay. So we use them as part of the suitability control in the T-cell assay for generic peptide project. So just to run you through the data set that we have generated doing the inter and intra variability analysis. So this is where we recruited a cohort of donors. We split their cells into 4 different sets, running them an independent study for each of the set. So this allows us to understand the inter and intra variability of us running the assay, and we validated this assay as well as submitted this data set to the FDA as part of the Type 5 drug master file. And this includes our full data package for the validation of the assay, as well as our detailed protocol, as we do understand that during the submission to the regulatory agency, these are questions that will come out that they need to understand how was this assay exactly validated, what are the data set, and what was the protocol that was performed as part of your submission for your generic peptide project. So then back to the point whereby we have to select a concentration. So this is also another challenge in terms of how do we then decide the optimal concentration. So this optimal concentration that we need to select, it doesn't impact cell viability. So typically what we recommend is that we run them at 5 micromolar, and then we can run the suitably control at 5 micromolar to test them alongside your impurities as well as your API. And typically how we perform the APC viability is through a process of elimination based on the characteristic of the phenotypic characteristic of these T cells. So where we focus on in the APC subset of analysis is looking at the B cells, monocyte dendritic cells, and CD4 T cells. And these are the key markers that we look at in terms of identifying this subset of APC through a flow cytometry method. So we have been talking about the analysis of peptide in a T cell functional assay, but what about testing a whole protein that is suited to understand about functional T cell assay? So just to take a step back, in the ProMap T cell proliferation assay, in principle, we would not recommend to test whole protein, basically because it's direct incubation to the PBMC. Even though the PBMC, they do have a level of APC, but we need to remember that it doesn't have a high level of professional antigen presenting cells, which is very critical part in terms of processing a complex molecule such as like a protein. So this is where we have, using the DCT assay that includes the incubation of the protein to the DC, which is the professional antigen presenting cells, allowing it to process the protein, the complex protein, into shorter peptides, followed by the presentation on the MHC class 2, followed by incubating to the PBMC for 7 days to measure the CD4 T cell proliferation. So typically how we perform this assay, we will recruit a panel of 20 to 50 donors, depending on which stage of the project you are at. We can make recommendation, and we will then have a global distribution frequency of the general population, and they're all fully class 2 type for DRBP-DQ. And the DCs, they are generated from monocytes, and then we culture and load the protein of your interest. And then the matured DCs, there's a washing step in between, so to prevent any crossover from the of your antibody to the PBMC. But do bear in mind that even though there's a washing step in between, there'll still be some residual antibody might go into the PBMC. So why I wanted to actually highlight this is especially for a T-cell engager project, whereby just a residual, depending on the potency of course, a residual of the TCE, the T-cell engager, getting into the PBMC, that could actually drive the CD4 T cell response based on the mechanism. So that could actually potentially mask the immune response that we are actually wanting to actually measure for the unwanted immune response, looking at the immunogenicity of your T cell engager. So in terms of T cell engager, we have other methods that we will recommend, where we actually perform a 2-stage instead, whereby we run a ProPresent first to identify where are some of these key peptides that we need to focus on, followed by running the T cell assay. So it's a 2-stage process where your antibody would not be interacting with the PBMC in this case. So then this assay allows you to capture immunogenic risk impact on PTMs, aggregates, and excipients. So this is an assay whereby it's allowing you to test the whole protein drug that you are working on, that you have developed. So this is a quick snapshot of the data that for the DC T cell assay. So what you're looking here in this number here, so this is the work we've done with Elias Morgan, that they want to assess the humanized shark antibody to understand what's the risk of the immunogenicity. So what we have here for these there are 4 candidates that they have sent to our UK facility, and followed by 2 positive control for the assay that we have. And you can see that they are way off the chart in terms of the response index, as it should, that we have a strong response from our cohort of donors. And then these numbers that you are looking at, this is called the response index. So how we actually calculate the response index is multiplying the percentage of donors that are giving you a positive response multiplied by the strength. So because we use flow cytometry to look at the strength of the CD4 T cell proliferation, we are able to measure what's the strength of that response. So by multiplying these 2 components together, we are able to holistically understand what is the level of risk of your protein drug that you are working on. So just a quick way in terms of understanding the data, how can we actually interpret for this case, is we can actually simply do a rankings strategy, where these 4 antibodies here, we can rank them from the highest response index to the lowest response index, with highest response index indicating a higher risk of immunogenicity. So from here, basically BB10 giving you the lowest risk of immunogenicity, allowing you to make the decision to take which molecule forward into your development program. So I personally like this slide that was presented by Dr. Mike Zimmer from Moderna, because he nicely captured the integrated process of, especially from ProImmune assay, in terms of how he have actually successfully de-immunized these bacterial proteases. So as we know that actually bacterial proteases, they are immunogenic. So the first question is that he wants to find out where are some of these regions of this protein that he needs to work on to de-immunize the sequence. So first, he deployed the ProPresent assay. So yeah, he is able to actually identify where are some of these regions that are immunogenic, and from there he can actually do engineering on it. So from here, doing a ProPresent, identify the immunogenic peptides, and then he do an in silico mutational scanning to see which are the amino acid that's able to allow him to actually make that mutation, and then followed by testing the peptides in the T-cell proliferation assay to see whether or not those mutations are they capable of driving a T-cell response, and then this allows him to select a list of candidate protein variant, and then he would then need to do the functional testing of these proteases, whether or not after the mutation, is he still retaining the function of these enzymes. Then once he have selected the range of enzymes that still retains the function, then he will run the final assay using a DCT assay by testing the whole protein, whether or not it has actually successfully de-immunized the sequence, which rightly so that he has successfully showcased in the data set, whereby the successfulness of this strategy, that showing that he has actually removed the T-cell epitope content, which reduced comparing to the wild type on the CD4 T-cell responses. So just to highlight some of these regulatory guidance that was published in June 2023, so the FDA recommends sponsors to consider the evaluation of immunostimulation hazards, and they highlighted that many assays exist. Like one of the questions was, there's a lot of providers out there and there's a lot of methods out there, and using CFSE, it's really the gold standard to characterize T-cell responses, and that is the basis of how we perform our T-cell proliferation assay using CFSE proliferation assay. So these assays will evaluate the ability of T-cells to activate and proliferate, and in some cases assess innate immune activation of APCs. So the very last assay that I'm going to be introducing is the MHC review peptide binding assay. That this allows you to rapidly discover T-cell epitope to understand what are some of the HLA restriction. So this helps you to eliminate in silico inaccuracy of in silico prediction. And it's an end-to-end service from us helping you to synthesize the peptide, to testing the binding against the set of HLA that you are interested in, testing the stability, and we're also able to perform a competition binding assay. So basically, this assay, we just need you to let us know what are the peptides you are interested in, what are the HLA allele you want us to test, and then we can do that for you, synthesizing to running the binding assay. So you do not need to ship any materials to our UK facility in order to run this assay. And then we have a proven track record of using this assay to identify T-cell epitope. There's a lot of publication using the review binding assay to identify T-cell epitope in a rapid manner. And also this assay then enables you to actually characterize and understand the risk of immunogenicity, of whether what are some of the peptides that is binding to the HLA at a very strong and stable manner, and that could actually potentially be your T-cell epitope that you need to focus on. So we have the broadest coverage of DR, DP, DQ. Basically, we cover the super types of these HLA class 2 molecules. And these are the molecules that we cover for class 1, and for class 2, we have the P, the DR, and the DQ, and we're also able to perform for mouse class 2. So this is the full range of alleles that we are able to offer for the review binding assay. So just want to wrap things up in terms of the assay that we are able to, that I have just gone through earlier. So Just to bring back to the very first point that I mentioned about the complexity of understanding our immune responses, it's not an easy task. So basically, we need to combine and rely on several assays or even products or even reagents for us to really understand the immune responses. So I think this gives you a very good overview of which assay is able to help you answer which sort of question as part of this immune response mechanism. So as you can see that the ProPresent antigen presentation assay allows you to understand natural antigen processing, the peptide binding to the respective HLA restriction, and also the T-cell epitope identification. The ProMap T-cell assay allows you to understand the T-cell function and also T-cell epitope identification. And the ProScern DCT assay allows you to understand natural processing and also the T-cell function. So what I want to actually try to say here is that each of these assays have its own benefit in terms of answering a specific question. So in order to choose what would be the strategy that you want for your program, is that we need to first understand what is the objective of your program, and we are able to actually make suggestion as to what would be the most appropriate strategy to help you answer the question that you have for your program. Right, this is just a summary of this my talk. So managing immunogenicity by design is essential to winning the drug markets of the future. And generation of key immunogenicity data allows you to differentiate your biologics from competitors and access to a wide range of specialist assays such as like ProImmune. We are able to offer you an integrated assay system to select which of the assay that to answer your question, so that these are being answered appropriately, enabling you to actually make informed and well decision to take which molecule down into the further down in your program. And with the experience of ProImmune and our team, we are able to actually make suggestion, recommendation of how can you get to your objective in the quickest turnaround time and also to how to reduce the overall risk of your project. So these are the molecules that we have tested to date. Probably there is more to it, but these are generally the molecules that we have tested before. Yeah, and I'm just going to put my contact details here, and thank you so much for your attention.